right. I'm Justine with Light Nurse Paula. Today I'm going to be talking about anatomy and physiology. This presentation was put together by Leanne Jones and Chuck Skinner. And it just goes over basic anatomy and physiology. Okay, science. So this is just dating back how far medicine goes. Ancient Egyptians depict various hieroglyphics that indicate that there was medicinal practices as far as that far back. Hippocrates, which the Greek physician, is the father. Aristotle came up with the scientific method. Galen, Roman physician. Text on Anatomy was the definitive work for 1500 years. Berzelius is the reformer, began modern research. Harvey has to do with blood flow, and van Leeuwenhoek invented the microscope. As far as humans, we belong to the phylum kingdom Animalia, the family of Hominidae. We're in the class of Mammalia, and our order is primates. We have unique physical characteristics, all humans do. We have a different size and development of our brain. Bipedal locomotion is potentially specific, opposing thumbs, vocal structures, and stereoscopic vision as well. The cell is going to be the functional unit of life. That's very important to know. So the cell is the functional unit of life. Here is a quick depiction of a cell. We'll get into that a little bit later on in the presentation. Tissues are aggregate of similar cells that perform specific functions. Organs are going to be aggregate of two or more specific functions. And then body systems are going to consist of various organs that have similar or interrelated functions. So it goes cells, tissue, organ, body systems. We've got different systems: integumentary, skeletal, muscular, nervous, lymphatic. These are all specific to humans. We have planes for reference. These are very important to know with various different things. The main thing being, whenever you're doing things such as CT interpretation or radiology reports, trying to distinct where an area is on a patient, as far as like if they have a laceration, you need to be able to explain those in more of a scientific manner. So the planes, there are three planes specific to us. The sagittal, which could also be called the medial plane, that's going to separate longitudinal, anterior, posterior. Then you have the coronal plane or frontal plane. It's going to be vertical. It divides the body into dorsal and ventral portions. And then we also have the longitudinal, I believe I just said that. So the three different planes for anatomic positioning, they're very important to know. And these are just pictures that depict various planes. So the way they're dissected is based on the plane that they're utilizing. As well, you have a horizontal axis, longitudinal axis, anterior and posterior. We need to know that as far as if you're trying to describe where a discoloration is or a laceration is, if it's proximal or distal to a particular a marker of the body. So is it closer to it or further away from it? Is it more coronal, which would be toward the head? Is it caudal, which would be down? Is it dorsal? That's going to be, think of a dorsal fin. So is it more posterior, or is it anterior? Is it toward the front? These are all terms that we need to be familiar with for practice. As far as body cavities. There are many, many body cavities that we need to know and what's housed in each specific cavity. So the cranial cavity, that's going to have the brain. That's where the brain's housed. The spinal cavity, of course, some of these are very obvious. That's going to be the spinal cord, and as well as the extension of the brain going down. You have your thoracic cavity noted. That's going to be the house where the chest is. So you'll have your heart, great blood vessels, some lymph nodes, the bronchi, things like that. And then you have your abdominal cavity, which is noted down further. That's going to be where your the abdominal cavity is going to have two different kind of abdominal and then pelvic. So that would be your abdominopelvic cavity. And they're going to contain stomach, liver, gallbladder, and then your pelvic region is going to have the bladder, reproductive organs, things like that. In 1932, Walter Cannon coined the term homeostasis, and that's going to be internal consistency. That's what all humans strive for. That's how you know if somebody's compensating or not. They're striving to correct something that's gone disarray in the body. So tachycardia is a sign that there is not homeostasis at the moment that the body's fighting to maintain homeostasis. Different things in the body that must remain homeostatic. You could have a pH imbalance. Typical arterial pH is going to be 7.35 to 7.45. That will be important later on as far as ABG interpretation. That's going to distinguish any kind of metabolic or respiratory acidosis or alkalosis, which is an issue if there is an imbalance. Bicarbonate, which is usually 21 to 28. 22 to technical about it, but that's also important as far as with an ABG, and it'll determine if static or not. Your sodium 135 to 145, calcium 4.6 to 5.2, your glucose 70 to 100. So another example of anatomy and physiology as far as maintaining homeostasis, an example of a clotting cascade. These are all mechanisms that are going to be utilized, utilized to maintain homeostasis in the body. So things like the clotting cascade, they regulate any kind of issues that are going on to keep the body in a homeostasis. Examples of insulin lowering blood glucose. These are all mechanisms that you have a cause and this is the effect. These are things in the body that keep it normal. So you want to keep it coming back and remaining constant. You don't want spikes in the blood sugar. You don't want drops in the blood sugar. So there are all these safe falls that are there to keep these things normal. As far as body chemistry, atoms, ions, and molecules are going to make that up. Different bonds are formed. We have covalent bonds. They're going to be formed by atoms that share pairs of electrons. These are the strongest type of chemical bond. Ionic bonds are formed by atoms that transfer electrons, and there'll be weak bonding. Hydrogen bonds, when a hydrogen bond to a negative atom, it gains a slightly positive charge, and it's weakly attracted to another electronegative atom. Acid donate hydrogen ions to a solution. Bases lower the hydrogen ion concentration of a solution. Organic molecules contain atoms of carbon joined together by covalent bonds. Nitrogen, oxygen, phosphorus, sulfur may be perfect cellular functions. Carbohydrates contain carbon, hydrogen, and oxygen at a 1-to-1 ratio. Lipids are organic molecules that are nonpolar and are not soluble in water. Triglycerides, phospholipids, steroids, and prostaglandins, those are going to be examples of those. This is a phospholipid membrane. In body chemistry, proteins are a long chain of amino acid bonds by covalent bonds. There are 20 different types that allow for great variability. There's a helical chain that can bend or fold, producing a second or tertiary structure. This variety allows protein to assume incredibly specific functions. So protein functions, hemoglobin is going to be your oxygen transport, myoglobin, oxygen storage and muscle, insulin, the regulation of metabolism, mucin is going to form mucus, blood GP produces blood type, and your lipoproteins transport lipids in blood. So cellular structures and functions, you have the cell membrane and the associated structures. There's a fluid mosaic model and cellular movement. Your cytoplasm and its organelles. So here are these. This is the cell structure that we were looking at previously that said we would go back over. So you have your Golgi apparatus, the mitochondria, the cell wall, and the nucleus. And these are all functions inside of a cell. Your microfilaments and microtubules create a microskeleton, which are going to help your organelles move within the cell, like little feet, little dusters that kind of push it around. Lysosomes contain digestive enzymes and eliminate structures and molecules inside of the cell. They also digest the food vacuoles inside the cell. Your mitochondria contain an inner and outer membrane that are the primary site of energy production in the cell. So they're the powerhouse, right? The mitochondria contain inner and outer membranes that are the primary site of energy production in the cell. That's the mitochondria. You have rough and smooth endoplasmic reticulum. The rough one is going to be for protein synthesis, and your smooth is going to be more enzymatic reaction sites. So they're going to store your calcium and skeletal muscles for potential reactions. Your cell nucleus, inside there you have your DNA. DNA consists of two long strands bonded together through hydrogen bonds. Your PC, if you remember those from any microbiology class that you took or biology class, adenine, thymine, guanine, and cytosine, and your RNA. Protein synthesis and secretion. Your messenger RNA enters cytoplasm and attaches to ribosomes. Each RNA bonded to a specific amino acid. Then you have your anticodons of the tRNA pair with the codons of the triplets, thus developing the sequence of amino acids. And that's just how those bonds are formed for your RNA. Proteins destined for secretion are produced and attached. Secretory proteins are moved from the rough endoplasmic reticulum to the Golgi apparatus, where they're further modified and packaged into vesicles. So we also need to know the difference between mitosis and meiosis. And here's a depiction of each. Your plasma membranes, it gives formal passage of materials in and out of the cell. Cytoplasm is a matrix for chemical reactions to occur. Your endoplasmic reticulum metabolizes nonpolar compounds, assists in protein synthesis. And the ribosomes, they're going to be the functioning units that synthesize protein. Synthesizes carbs, package molecules for transport, secrete lipids and glycoproteins. The mitochondria release energy from food molecules. Lysosomes digest foreign molecules, invaders, and damaged cells. And they're going to be the oxidative reactions. Centrosomes organizes spindles during mitosis. Vacuoles store and excrete within the cytoplasm. Fibrils support cytoplasm and transport. Cilia and flagella move particles. Again, those are those things I was talking about, like kind of like if you think about an octopus, the way they move, the little legs that just move particles. Your nuclear membranes, those control passages into the nucleus. The nucleolus forms ribosomes, and then your chromatin controls cellular activity for life process. And that is the end of your basic anatomy biology level.